So indeed, I'm doing my PhD at the Rabat UMC within both the Amalia's Children's Hospital as well as the Human Genetics Department within the Rabat UMC. And today I'll give an update on my project, which is a personalized ASO screening for a Stargardt disease patient carrying a deep intronic ABCA4 variant. And before we do this, I'll give a quick introduction on the eye and specifically the retina. So the retina is a highly organized structure. I see, wait a minute. I see the top bar from the Zoom. I don't know if I can remove this because I cannot see the title of my slide at this moment.
We can see the title of your slide.
Okay.
Then the retina is a highly organized structure.
Yeah, okay. Then the bar is only visible for me at least. Okay, that's good. So yeah, like I mentioned, it's a neural tissue located at the back of the eye. It's a highly organized structure with distinct layers of cell types. Most importantly for today's topic are the photoreceptor cells in the photoreceptor cell layer. There are two distinct cell types. One is the rod photoreceptor, which are mainly located in the periphery of the retina and are important for low light vision. And then we also have the cone photoreceptors, which are located in the center of the retina and are important for color vision. These photoreceptors are responsible for the conversion of light into an electrical signal, and this happens at the photoreceptor outer segments. And from this conversion, it's transferred into other neural cell layers to the ganglion cell layer, and this signal will then be sent to the brain where it's processed for visual function. These photoreceptors are in contact with epithelial cell layer called retinal pigment epithelial cells. And together, these RPE cells, together with the vasculature, form the blood-retinal barrier, which is similar to what the blood-brain barrier is for your brain. And these cell types, so the photoreceptor cells, but also the RPE and the vasculature as well, are the main cell types that are affected in retinal degeneration, the topic of today's talk. Specifically, I'll focus on inherited retinal diseases, which has a prevalence of about 1 in 2,000 and have an early onset. But there's a large group of inherited retinal diseases, each with their own disease phenotype, whether it's progressive or not, and whether there's more rod involvement, such as retinitis pigmentosa, or whether cones are more affected, such as in Stargardt disease. So it's a group of disorders, making it quite heterogeneous. But not only that, also on genetic level, inherited retinal diseases have high heterogeneity, with a lot of different genes affecting a lot of different phenotypes. So today's talk, I'll only focus on Stargardt disease, and specifically Stargardt disease type 1, which is kind of the exception because this disease is caused by variants in the ABCA4 gene. So Stargardt disease is a macular degenerative disease leading to the degeneration of both the cone photoreceptors as well as the RPE cells within the center of the retina, the macula. So in the bottom, you can see a fundus image of an eye. Within the center, there's the macula. And in Stargardt disease patients, we see degeneration. And this leads to progressive vision loss. Stargardt disease is estimated to affect about 1 in 10,000 people globally. And although Stargardt disease type 1 is caused by variants in the ABCA4 gene, over 2,800 unique ABCA4 variants have been identified, from which an estimated 1,800 are classified as pathogenic or disease-causing. Because there are so many disease-causing variants in ABCA4, there's also a lot of patients with ultra-rare variants or even N of 1 variants. So one patient carrying one specific disease-causing variant. And this gives the opportunity for N of 1 trials for personalized RNA therapies. The most famous story or example from this is the story of Mila, where the group of Tim Yu at the Boston Children's Hospital designed a patient-customized oligonucleotide therapy for Mila, a girl with Batten's disease. And the great thing is that although Mila unfortunately passed away in the end, this was quite effective as Mila saw a reduction in the number as well as the severity of her seizures that she had. Within the Netherlands, we're trying to set up a very similar thing with the Dutch Center of RNA Therapeutics, or the DCRT. And this is a non-profit consortium between 3 academic centers within the Netherlands. So we have the Leiden University Medical Center, the Radboud Medical Center in Rotterdam, the Erasmus Medical Center in Rotterdam, and the Radboud UMC in Nijmegen, where we are working. And the aim of the DCRT is to develop these tailor-made RNA therapies for patients with ultra-rare genetic mutations, focusing on the eye and the central nervous system, and to develop these therapies in-house and to provide them to a patient at cost. Within the Radboud UMC, we identified a Stargardt disease patient that might be eligible for this N-of-1 type treatment. There are several criteria. One is that the ASO, or the antisense therapy, must restore protein function, that the patient has a good and predictable prognosis, and that there's still a therapeutic window for the therapy to have an effect. So this patient that was identified is carrying two variants within the ABCA4 gene, both of which are located deep intronic. The first variant, which I like to call the 1137 variant, is a more common variant and therefore not suitable for this N-of-1 trial. But the other variant, the -506 variant, is an ultra-rare variant and is therefore a target for our ASO screen. And I say that this is an ultra-rare variant and not an N-of-1 variant, because there in the past are already some patients identified with the same variant. For now, we know that there are at least 4 patients carrying this variant, so still there are very few patients that are affected by this variant. But at least this leads to already some preliminary work being done on this variant and helps us develop a therapy for our specific patient. And the -506 variant is located between exon 7 and 8 of the ABCA4 gene, and this variant causes an inclusion of a pseudoexon in the transcript. So normally what happens with the protein synthesis pathway is that DNA is transcribed into pre-mRNA consisting out of exons and introns. Through splicing, these introns are removed, creating mRNA, and this mRNA is translated into a protein. And when we try to visualize this in cells, we can do a reverse transcript PCR, and we nicely see one band appearing. In this case, however, part of the intron is recognized as a pseudoexon. And instead of being spliced out, this is included in the mRNA transcript. The mRNA transcript is then degraded, leading to no protein. So when we try to visualize this in a Stargardt disease patient, and as I mentioned, this is previous data, as there were already a few Stargardt disease patients carrying this variant identified in the past. We see that when we inhibit nonsense-mediated decay using cyclooxamide, we see a second band appearing, the transcript containing the pseudoexon. Also, in the past, several ASOs were already tested against this variant, and some of which seem to be effective in restoring the correct transcript expression. So from our patient, the first thing that we did is indeed confirm that this patient is expressing the pseudoexon, which after we inhibit nonsense-mediated decay in the cells of this patient, indeed seems to be the case. So then our strategy is to work with 4 different ASOs, and these ASOs all have the 2-MRE modification and fully PS backbone, to use these 4 ASOs to find the most effective one for our specific patient. But before we can give this ASO to the patient, we of course have to test for efficacy. And normally in normal drug development, you might use an animal model. However, in our case, these animal models are not suitable as, well, these ASOs require a sequence-specific approach, but also it doesn't make sense to create a humanized model carrying this specific variant if there's only one patient. It's just way too expensive and costs way too much time. Alternatively, conventional cell models are also not suitable, as we require a tissue and cell-specific expression. And ABCA4, a gene of interest, is mainly expressed in the photoreceptor outer segments in the retina. So here you can see a quick schematic of ABCA4 expression. And ABCA4 is mainly expressed in the outer segments, in this part of photoreceptor cells, and acts as a transporter of visual cycle products. And this is a very simplified version of the function of ABCA4, but in reality, this is way more complex. But since I won't talk about the pathway or disease pathway of ABCA4, I will not go into more detail about the function of ABCA4. So if we want to have both a sequence-specific approach as well as a tissue-specific approach, our solution for this is using iPSC-derived models or induced pluripotent stem cell-derived models. And we're also using these models to do some initial safety testing of our top candidate ASOs. Our iPSC model of our choice is retinal organoids, and these are three-dimensional tissues that resemble the human retina. They're usually about 2 millimeters in diameter. And as you can see closely, they have what we call a brush-like border around the edge of the retinal organoid. If you zoom in a little bit more, you can see it a bit more clearly. And when we do an immunofluorescence, we see that they have a highly laminated structure as well, which is very similar to the structure of the human retina. We can create these retinal organoids from iPSCs. I'll go over the very basic protocol very quickly, where we generate embryo bodies. We can then plate these embryo bodies sparsely on a 6-well plate and allow them to grow for over a week. Then we again scrape them off and allow them to self-aggregate. And here at day 21, we can already see very laminated structures that resemble the retina in retinal organoids. But from this, they're still very immature, and we have to mature them before they get a brush border, which takes over 150 days before they express these photoreceptor outer segments. The great thing about this organoids, though, is as we do wait this long, we see expression of ABCA4 protein, so our protein of interest. So here you can see an immunofluorescent image of day 156 retinal organoids. And in a Stargardt disease patient, we see a highly decreased expression of ABCA4. So we know that we can detect ABCA4 protein after about 150 days in vitro, which to us is a very long time, and I hope for you as well. But luckily, we did some preliminary work on retinal organoids with using qPCR, and we see that the gene expression of ABCA4 already increases in earlier stages. We also confirmed this with publicly available RNA sequencing data of developing retinal organoids. And from this, we estimated that after about 50 days, there's already at least a 10-fold increase of ABCA4 gene expression, so even before protein expression starts. So we made use of this to test the efficacy of these ASOs on gene level. So what we did is from our N-of-1 patient, we got patient fibroblast. Our stem cell facility at our hospital created iPSCs from this. We then generated retinal organoids, and at day 40, we started the ASO treatment. So for this initial treatment, we used 5 micromolar of ASO, and we did a 10-day treatment. The day before you harvested, we inhibited nonsense mediated decay using cyclophosphamide to again see all the transcripts that are present. We then did pseudo-exon skipping analysis using RT-PCR, as I showed you before. So here you can see an overview of our results. So if we do this on control organoids, we again see nicely one band. And in a Stargardt disease patient, we see a second band appearing. And when we add cyclophosphamide, so when we inhibit nonsense mediated decay, we see an increase in the intensity of this upper band. So this pseudo-exon included transcript. We also added the scrambled ASO as a control, and here we see the very same thing as the non-treated sample. And we added our 4 different ASOs, and we see various degrees of correction of the pseudo-exon. Then we calculated the ratio between the pseudo-exon included transcript and the correct transcript, and we saw that specifically ASO3 and 4 are highly effective in restoring normal transcript expression. We then continued with these top 2 ASOs, so ASO3 and 4, to also look on protein level. So what we did, this is organoids further developed, and at day 175, we started again a treatment. This time we did the treatment for 30 days, again with 5 micromolar ASO, to look at the recovery of ABCA4 protein using Western blot. So here you can see the results of Western blot. So compared to a control retinal organoid, we indeed see a huge decrease in ABCA4 protein, as was expected in this patient. The scrambled ASO did not help increasing the ABCA4 protein expression. And our 2 ASOs we added here, you can see various degrees of increase in ABCA4 expression. So when we calculate the percentage of ABCA4 expression, we see that only for ASO3, we see a significant increase in ABCA4 protein, making this our top candidate. So apart from looking at efficacy, we also wanted to use retinal organoids to look at safety. So for this, we did 2 different assays. So we treated day 200 plus retinal organoids with this time 10 micromolar of ASO, which is twice the concentration that we use for our efficacy test. And then we harvested the retinal organoids for a cell viability assay using cell titer glow. And for the media, we collected for a cytotoxicity assay using a lactate dehydrogenase assay. So to look at cytotoxicity, here you can see the results for our non-treated compared to our 2 ASOs. You can see that nothing much is happening. We also included a sample that we treated with ultevursen, which is an ASO that is now being recruited for phase 3 for Usher syndrome. So this is already deemed clinically safe in patients. And also here, we did not see any difference between our ASOs and the ultevursen. We also included 2 positive controls. One we treated with doxorubicin, which caused cytotoxicity, and the other ones we lysed with 10% triton to get an idea of the max LDH of these organoids. And also here we saw an increase. We used the same samples for our cell viability assay. And also here we did not see a decrease in cell viability in our 2 ASOs of interest, but also not ultevursen. And for doxorubicin and max LDH treated sample, we see indeed a massive decrease in cell viability. So this is the work on safety that we did in retinal organoids. We also did an initial safety testing in peripheral blood mononuclear cells, or PBMCs, where we stimulated PBMCs for 24 hours with our ASOs and checked for the release of pro-inflammatory cytokines. And this was done by a technician in our group, Femke, and a former postdoc in our group, Dia. So after we stimulated them, we measured pro-inflammatory cytokine release using an ELISA. And you can see an overview of the results here. This is a lot of information at the same time, so I'll quickly break it down for you. In this table, each row represents a different pro-inflammatory cytokine. On the right, we have 2 positive controls, R848 and the CpG ODN, which are known to elicit a pro-inflammatory response. We then again included ultevursen, the clinically safe ASO. And then we used here 3 different ASOs in 3 different concentrations. And overall, we did not see a massive increase or major immune responses after ASO treatment in PBMCs. So I want to summarize what we found so far. So we used retinal organoids as a model to test ASO efficacy. Here we found that ASO can restore wild-type ABCA4 gene expression, and specifically we saw that ASO3 and 4 seem to be highly effective for this. And we also saw that ASO3 seems to be effective in restoring ABCA4 protein, making this our top candidate for now. For safety, we use multiple in vitro models to test ASO safety. So for ASO3 and 4, we did not see an increase in cytotoxicity using the LDH assay or a decrease in cell viability using cell titer glow in retinal organoids. And ASOs 3 and 4 did not elicit any increased inflammatory response in PBMCs. So currently, there are still a few things that we want to do in our lab. One is to also check with immunofluorescence on ASO-treated retinal organoids. We're currently in the process of performing these experiments where we not only want to see whether there is an increase in protein, but that this protein also goes to the correct location, which will be the photoreceptor outer segments. And we're setting up a safety study in rabbit as well, just to see if we see any adverse events. So this is some stuff that's still ongoing. But before I can give it to the patient, there still has to be quite a few things that need to be done. One is the GMP manufacturing of the ASO, because for now we used experimental quality of ASO and not GMP quality ASO. We also need to get approval from the regulatory bodies to start this treatment of this N-of-1 patient as an experimental treatment. And also, we still need to acquire funding for the treatment itself. For the research parts, we do have funding, but for the treatment itself, we're still acquiring funding. So this was not only the work for me, but a lot of people, especially people in the group of the blindness genetic therapy, with my supervisors, Rob Collin, but also Alex Garanto. I already highlighted some technicians, Femke, but also Anita, Lonere, and Leonie were of massive help. So at the Human Genetics Department, Frans and Susanna for identifying the variant, and ophthalmology, of course, for actually seeing the patient, and the DCRT for setting up this whole consortia to make N of 1 treatments possible within the Netherlands. And lastly, I want to thank you as well as the organizers for the opportunity to to present my research here, as well as at the OTS last October. Thank you.
Yes, thanks a lot, Edwin, for this great presentation. Really enjoyed it. So a few questions from my side. And if you have a question for Edwin, please use the Q&A function. So first question from me. Can you tell us something about the disease progression in this one patient? So I think it's always important to have a little bit of the background. So how progressive is this disorder? And how can you measure whether treatment works, yes or no?
Yeah, right. That's a very good point. So actually rates of progression I cannot give because I'm not an ophthalmologist, of course. But we have been closely monitoring this patient at the ophthalmology department, getting the natural history of this patient of the disease progression, to try to get as big of a picture of how this disease is normally progressing in this patient. We have now done this for the last, I think, year and a half, 2 years maybe already. So we have a nice idea of how this disease is progressing in this one specific patient. So during this treatment, if we see that this is diverting, it also gives us an idea that the treatment is effective. Yeah.
And then I have a question about your treatment, because I think somewhere in the introduction you mentioned that you are treating ultra rare variants. So I think this particular patient has the -506 variant, if I wrote it down correctly. But you also mentioned that this patient has a very common variant, the 1137. And to me, it sounds a little bit counteractive that you are going for a treatment for an ultra rare variant. Well, you could also treat a bigger group of patients while developing a therapy for this very common variant. So can you comment a bit on that?
Yeah, well, you're absolutely right. It does make sense to start treating as many patients as possible, of course. So also for this 1137 variant, there are treatments in the works, especially in the group of Rob Collin and also his spin-off company. So that's definitely in the works. But for now, we're focusing also on the N of 1 trial to show the regulators as well that we can do this and that ASOs are a safe and viable treatment option for patients. So if we focus on the N of 1 patients, it might also help other therapies, other ASOs for more common variants to be accepted as a treatment in earlier stages.
Yep. So for the efficacy of your oligo, you use the retinal organoids. And if I listened correctly to your presentation, you mentioned like 150 days, 175 days, 200 days. So to me, that sounds quite some time. And I've seen that, for example, I think within your group, you also develop different kind of mini-gene assays or mini-gene assays. First question, would that not be enough? Just to use a mini-gene assay to see whether splicing can be corrected?
So this mini-gene assay was indeed developed for this variant in the paper that I also quickly discussed with the preliminary data. So they did create a mini-gene for this variant. But the issue with the mini-gene is that you only have very little, a very short, medium part of the ABCA4 gene and not the whole genetic context. So that makes it difficult. One, we don't know if the splicing is the same in a mini-gene as it is in, the patient or retinal organoids. We know that for some other variants, it's definitely not the same, where we don't see anything in, for example, mini-genes, but we do see it in iPSCs derived retinal organoids. And also on protein level, you cannot really see anything in mini-genes as well.
Yeah, and then the follow-up question, I think one other advantage of the iPSC-derived models is that you can do some functional assays. So are there any functional assays with retinal organoids? Can they, I don't know, give it something with light that they can see, or how does that work?
Yeah, so this is very much still well in baby steps. We're trying to set these things up for inherited retinal diseases. So indeed, we're trying to see whether there are changes in light responses, but as well as in metabolism. Because as I mentioned, ABCA4 is important for the transport of visual cycle lipids. So we're trying to see if there's a difference in lipid metabolism, for example, in retinal organoids from a patient compared to controls. But we're very much still setting this up. So before including this in ASO efficacy testing, we're still quite far from that, I think.
Yeah, that nicely moves to the toxicity testing. So let's see, we have a few questions in the Q&A. So based on your experience, would retinal organoids inform retinal degeneration if there's any caused by the ASO treatment?
So what? Okay. So the question is whether there would be retinal degeneration if we treat them with the ASO that is potentially toxic.
Yes.
Yeah. So this is something for one of the toxicity assays. So for the LDH assay, we also collected media one day after we treated. And there, when we treated with, we only did this for one replicate, so I did not include it in this presentation. But when we included the CpG ODN, which is more similar to what an ASO would be, we did see an increase in LDH in these organoids. After one day, after day 10, we did not see anything. But after day 1, we did not see any effect on LDH in our retinal organoids, on the ASO-treated retinal organoids.
Yeah, so
massive degeneration. We have not detected this. So ASO mediated degeneration. I'm not 100% sure. Yeah.
So I know you're working on an N-of-1 case, so I might assume that toxicity testing is still very important, but can be done on another level, using, for example, your retinal organoids. So are there any papers known, or do you know anything about the comparison between the tox test in these retinal organoids compared to the traditional tox test in the animal species?
That's a very, very good point. I'm not aware of these patients or how reproducible these results in retinal organoids are compared to animal models. This is also definitely why we are including an animal model as well. So we think that the ASO is safe, but of course we are going to test this also with an animal model because we cannot be more sure than we are. So that's one part of the question. So we are not 100% sure. So just to be sure, we're also including an animal model. But at the same time, we also don't know how well the translation from an animal model is to the human situation, right? So yeah, there are two sides to this point. Yeah, but we can never be too sure. So we definitely, opening all options to make sure that it's safe to give to the patient. Yeah.
Last question from the chat. It's about the distribution in the eye after the IV injections. Do they distribute well in the retina and the cornea?
Yeah, so I know that also in our group, but also in other groups, they've done an RNA scope on these ASOs to see the distribution of these ASOs, and they seem to be well distributed with intravitreal injection. Yes.
Okay, well, thanks a lot. I think I give the word to Erin.
Yeah, so our next speaker is Trisha. Perfect, so we can see...
Hello.
We can see
Can you hear me as well?
Yeah, we can see your slides, but they're still in like the overview. There we go, perfect. Go ahead.
Hello, everyone. My name is Trishalina Das, and I'm a PhD student at McGill University in Prof. Hanadi Slima's lab. Today, I'll be talking to you about some preliminary findings from our lab on exploring nucleic acid data assemblies at delivery vehicle. So this audience does not require an introduction on how nucleic acid-based therapeutics work and how they have revolutionized the field of medicine. They can help at each step of the central dogma, from DNA to RNA to protein levels, and can either upregulate or downregulate a gene or a product of interest, and just help in curing a disease. In our lab, we essentially use nucleic acids as therapeutics, but also as structural material. So, based on the highly programmable nature of the DNA and RNA, we can essentially make different shapes and sizes and structures from it, and then precisely control these sizes and shapes because of their highly programmable nature. In our lab, we essentially work with two different kinds of DNA and RNA assemblies. The ones which are on the bottom are just made of pure nucleic acids, and they are known as wireframe structures. The ones that I'll be mostly focusing on today are the spheres, sheet, and fiber. I'll focusing more on the spheres. These are essentially made of different polymers. So where we take advantage of and integrate autoclave interactions such as hydrophobic effects in order to form nanoscale assemblies of different shapes and sizes, which can be used as delivery materials. So for the rest of my talk, I'll be focusing on the spherical nucleic acids, which are made up of sequence-defined copolymers. So if you look at how they are formed, these are self-assembled. And just when you do a DNA synthesis, we can essentially add monomers one after the another during the synthesis. In place of a nucleic acid monomer, you can also add something known as a non-nucleic acid monomer. And in our case, this is a dodecyl alkyl. This can be added just like a normal monomer and can be processed just like that. So we can add one after the another. For our studies, we're essentially adding 12 of these dodecyl alkyls, and then after they're cleaved and separated from the support, we essentially have something like this, where we have a sequence-defined polymer with a hydrophobic nucleic acid portion and a hydrophilic nucleic acid portion and a hydrophobic rock portion, which can then assemble to form the tracene. So till now in our lab, most of the work has been done in vitro. So these are so my work will be essentially looking into how these some structures behave in vivo. So if you look at the structure itself, the design, as I said, is of a phobic chain, and then we have the RNA portion. For our studies, the RNA for studies and nucleic therapeutic, we'll be using siRNA, and this siRNA has been used by the before for targeting Huntington. Also, we have an RNA portion and a quick chain here, and then we have and this is for this RNA. This can then essentially assembled in to have these structures where we have the hydrophobic core in the inside and the RNA in the outside. For my studies, so we have nucleic acid corona, and then we have hydrophobic core. For these studies, we'll have essentially two kinds of cores, where in one case, we have the normal soft backbone, and in the other case, we just have the replacement of one of the oxygen with sulfur. So these are the two structures that I'm mostly focusing on for my talk. So after the design is done, so after we essentially assemble them, we can then check their assembly in a gel. So we run a 2.5% native gel, and we can see that they have been forming pretty well. The first one is just the structure without the antithesis part, and the second design is with antithesis portion to it. Then look at the muffler. Firstly, we use AFM, where we can see that there are actually these dots which are spheres, and then look at the size with DLF analysis, where we find that they are 20 meters in size. So now we have this SNEs with a bit more in the inside and the the outside. So we have these structures which have been assembled characterized successfully. After this, we go first, we did some studies with our collaborators at the LDI. So with them, we did some blood chemistry as well as CBC studies, and we found that the structures, when they're injected by IV injection on day 7 at 10 mg per kg doses, they essentially are not that toxic, and they can compare to the buffer control, which is in blue here. So we have different structures here, over here, and then when we weigh the mice over the period of 7 days, we see that their weight doesn't fluctuate much. So we can see that based on this short study that our structures are not toxic, and then we can move to the next studies. So in the next part, what we essentially did was looking at the biodistribution of the structures. So just one where we have the PO core, we can see that they give the liver, which is like bottleneck nanoparticle distribution is a big go to liver, but we are very happy to see that we also see some lung accumulation as well as some heart accumulation. For our PS, where we just have one oxygen replaced with sulfur, we see that it goes to the kidneys, which means that we can, just by changing a very small part of the core, we can essentially have different which is actually very interesting for us because we play with the core a lot. All of this was done using IV injections, and we image the structures after 48 hours after we sacrifice the mice and look at each of the organs of the mice. So we set on day 7, and on day 7 is mostly extracted out of the body to the of the body. So with this in mind, since we want to check how good they are as an look at the relative mRNA expression of the Huntington gene, the Huntington mRNA, which is essentially we just express in the past body. We focus on some of the important body, the liver, kidney, skin, lung, heart as a bone marrow cells, and we see some amount of silencing in this its organs. These are all done by using FBB mice and IV injections on day 7 with a 10 mg per kg dosage. So now the party said, since we saw this we should change the core a bit, can you see this also in the silencing as well? So we also see it a bit in the silencing as well. The PS4SNE does not silence as much. The results out here are not that significant, but they're very promising because this is our first foray into seeing how this structure behave in vivo. So in the end, we will be essentially moving with higher process and also play with the analytical chemistry to make it more efficient. So at the end, in conclusion, we had siRNA SNEs which can be assembled. We also saw that they're localizing organ for the liver, and we also saw that they had looked in the future work. As I already mentioned, you want to optimize the chemical modification of the siRNA to have better efficacy. Also will increase the dosage to see if we can get better response. So with this, I hope I was able to tell you a bit about this area of research. And then I will thank my PI and OTS for giving me this the talk here. Also, my contact details are here. So if you have any questions, you can also mail me there. So thank you, and any questions and really welcome. Thank you.
Yes, thanks a lot, Trisha. Wonderful presentation. So also for Trisha, if you have any questions, please use the Q&A function. So maybe very first question, Huntington disease is a brain disorder, and I think you tested a lot of different organs to check the biodistribution, but I did not see the brain. So did you also test the biodistribution in brain, or is there any of the compound reaching the brain?
We did not do that because the collaborators that we are working with, they essentially were not not that. So we did not think of looking at the brain because our main motive was just to look at where our structures are going. So our next part of the experiment will also look into the brain, but currently did not look in the brain as such.
Okay,
So that's a pilot study.
Yeah, it would be super interesting to see what's going on there, because also I'm not super aware what the expression levels are in all the different organs. So do you think that it matters what the expression of a particular gene is in a particular organ? Can be, for example, can be very highly expressed in organ A, and maybe you see like a slide effect. Well, it's hardly expressed in another organ, and then it might be much more reduction.
Yes, that might be a point that we have to keep in mind going forward, because then we have to interpret data based on that. So we'll be looking forward. We'll be going to look at that as well. But since it's already been used by another lab to do the same kind of studies, where they did not find any difference, so we were just going with that based on that study.
And you showed some very nice data with the HTT siRNA with the PO core, and the other one was with the PS core. And you do see some promising results using the PO core, less promising results using the PS core. So can you explain these differences? So what is it in these cores that makes it different? Do you have any idea?
So the SNAs that we have are essentially will be something like this, where we have the hydrophobic portion in the inside and the siRNA on the outside. In our cases, the PO core, we have the phosphate link. PS core has just one of the oxygen replaced by the sulfur. Previously, in our research, we have seen that HTTPS that they're essentially monomers, something like this, whereas the HTTPO is essentially still a particle. So based on that, our results essentially shows that maybe if it's a particle is more efficient as compared to being just monomer by itself. So that is one of our main ideas.
Also, another obvious question. So what would be next?
So this is a very small experiment. So we want to test different cores. Our lab has a lot of technical expertise in developing these cores. So since just by changing a very small oxygen to sulfur, we are seeing this difference. So we are going into more different core chemistries, essentially with the same siRNA, and also because our results here are promising, but they are not here. So we'll be still changing the pattern of the siRNA to check if we can have more efficacy as well later on.
Okay, well, thank you very much. And I give the word back to Erin.
Great. And so our last speaker for today is Josh.
Yeah, share my screen. Cool. Hey everyone, I'm Josh Robinson, a PhD candidate from UT Southwestern and Daniel Seeger's lab. And today I'll just give you an update on the work that we're doing on my project right now. We're doing a lab titled Novel 3-Component Zwitteronic Aminolipid Nanoparticles for mRNA Delivery. So to begin, LNPs are the most clinically advanced delivery system going back 2018 alnylam's Onpattro. It was used to deliver siRNA, but as you see here, there are 4 components, and these 4 components are pretty standard across most LNPs. You typically have a ionizable lipid. In this case, it's D-lin MC3-DMA. This is really important for actually interacting with the genetic cargo. You also have a phospholipid helper lipid, which is also important for the interaction, and also stability of the LNP. Cholesterol is important for membrane fluidity, and you have PEG 2000 or polyethylene glycol lipid that is really important for extending blood circulation and also preventing aggregation. So fast forward 2019, 2020, we have Moderna and Pfizer and BioNTech with their COVID-19 vaccines. As you see, again, we have those 4 components. However, you see a little bit difference in the ionizable lipid. So as you see here, there's actually now an inclusion of branched allotales. And more important for my project, we have the inclusion of hydroxyl group. So these are 4 components. Typically, they're 4 component LNPs, and typically they go to the liver. However, our lab, a little earlier than that, by 2017, we wanted to kind of figure out, is there a way to actually combine 2 components into one to actually make a less complex LNP and also have increased molecular interactions with longer messenger RNAs as opposed to siRNA of the cargos. And we were able to do this. We combined aspects of ionizable lipid and also the phospholipid, and we termed them zwitterionic aminolipids, or ZALs, as you see here. So with the ZALs, we did a pretty large screen, and we were able to find our top candidate called ZA3-EPI10. That's the structure you see here. You see also that it has extra hepatic delivery, which is really nice to see, as a lot of LNPs typically are go to the liver. So this is actually the second part that builds up my project. So we have the hydroxyl group and we have ZA3-EPI10. So what is my project? So with both of those aspects in mind, we looked at actually seeing if we can vary the chemical space at the hydroxyl position. As you saw that Moderna and Pfizer had the hydroxyl position. And we also wanted to use ZA3-EPI10 to make a 3-component LNP as opposed to a 4-component LNP. So with that, I was able to synthesize these derivatives you see here, kind of varying the chemical space. We have the inclusion of the halogens with the chloride and the bromide, and also showing the more bulky side of chemical modifications. We have the opivylate, as you see here, and then we have the acetate, which is kind of more in between with the hydroxyl group. So with those derivatives in hand, we went ahead and actually formulated them into 3-component LNPs. And these are the 3 components. You have your ZAL, PEG, and cholesterol, and then the mole ratio and mole percents. And from there, we did a pretty standard physical characterization, looked at size of PDI, and this is kind of where we're kind of starting seeing some degrees of change. So in the case of all of the LNPs, we have PDIs below 0.2, which is really nice because PDI points to homogeneity of the LNP. So essentially, a low PDI shows that in general in that solution, all the LNPs are about the size that you're seeing. So looking at size, we see that the acetate is pretty roughly the same size as the hydroxyl group. However, we see from all the other derivatives that there's an increase in size, with the opivylate being the biggest and the halogens with being really electronegative, they're also bigger as well. Then we're going to look at mRNA encapsulation. We look at, in my case, there are multiple cargos, genetic cargos, of course, but we're looking at mRNA. See across the board that all of the derivatives and also the hydroxyl ZA3-EPI10 have really nice encapsulation. That's good to see there. We then wanted to look at apparent pKa. This is a little more new. Our lab was able to correlate pKa, but also organ fate. And typically, LNPs above 9.25 or more lung targeted. Between that 6 and 7 and 6.4 is kind of like optimal range for liver. And then below 6 is more spleen targeted. So as you see here, all the LNPs made actually are below that 6. So we hypothesized that based on physical characteristics, they'll go to the spleen. And then we want to look at zeta potential. For the most part, all of the LNPs are fairly neutral. This is really just to kind of really give more of the differences, but zeta potential will go from minus and plus to minus 50. So as we see, it's compared to plus or minus 50, fairly neutral, as we expect, because there's not really any positively cationic or anionic elements in the LNPs. So with the LNPs made and fully characterized, we went and wanted to do initial in vitro work. We did this in iGrov1 cells. We see here that there's really high cell viability across the board, which is really great. However, we actually looked at the in vitro mRNA transvection. we see that the parent performs well as expected from previous results. However, the acetate this still performs not nearly as high, but it's definitely there. The other derivatives do not really perform. So you still want to go ahead, even though we have the in vitro, we wanted to go do in vivo because oftentimes we see that in vitro and in vivo don't always correspond, typically due to just the fact that there are really complex biochemical systems in mice as opposed to just in cells. So we went ahead and did in vivo, and sure enough, we did see a difference. So in the case of the bromide chloride pivolate, there's still no delivery there, which is fine. That kind of correlates to in vitro. However, looking at in vivo, we see a stark difference. So we see that the acetate actually has really high delivery. And not only does it have really high delivery, it's also really high in the spleen. That is correlated to pKa. And also the parent also has more spleen target, so pKa dependent there. So that's good to see. But the acetate is much higher. And then if we actually look at this, just looking at just the spleen, you see there's almost a twofold difference between the parent and the acetate. And then we're looking at the other derivatives, there's not much there, which is fine. Then also we look at functional organ distribution. We see that for the most part, parent, acetate, and pivolate, all are spleen targeted as we would expect from pKa. However, we saw that the halogens do not really hold to that, and they have a little different tropism in that regard. But we see that the acetate not only performs the best, also has really high and the highest spleen specificity. So from there, we see that we have at least one new derivative of the acetate that works well. And we did further work without the other derivatives as there's not much delivery there. So what more did we do? We wanted to actually look at some engineering. So we actually took the acetate and the parent, and we changed their PEG content. So there's only 3 components in these LNPs. The ZAL has obviously changed a whole lot. We also have cholesterol, which is fairly studied in the field as far as what changes happen when you modify it. It wasn't too much looking at PEG, and also PEG is in such a low percentage. You're looking at around 0.5%. Typically, LNPs are around 2 to 5%, so it's already very low. So we're hypothesizing that little small changes might make some big changes happen. So for the most part, it didn't really alter anything. Encapsulation stays about the same across the board. pKa is changed some, but it's still around that spleen targeting range. And then zeta potential is fairly neutral still. However, previous data had actually shown from other groups that if you change the size, you change the PEG content, you actually see a size decrease. And what's really nice is that we did see that. We did see that as you increase PEG, you decrease the size across the board. So that was really nice to see there. So with this in mind, we have the physical characterization. We opted to actually skip in vitro and go straight to mice. And we see that actually PEG change does make a big difference in mRNA delivery. Looking at the hydroxyl group or the parent Z3AP10, we see that there's more of a highest at 0.25% PEG. However, when you're going up to 2%, we see a decrease that corresponds with that. However, in the case of the acetate, we see that there's more of a peak. Looking at that more quantitatively, we see that definitely happens here. As the parent compound, as the PEG goes up, activity goes down. But what's also interesting is that as PEG goes up, we actually see that there's more of a detargeting that's not as specific to the spleen. However, in the case of the acetate, not only is delivery the highest overall, we also have a really nice retention of spleen specificity. So that's really nice to see there for a new derivative, which is great. So what are our key findings? We see that the ZAL scaffold is really nice for doing chemical modification to optimize and enhance delivery efficacy. We see that the acetate modified Z3AP10 is more efficacious than the parent and also more specific. And we also see from an engineering perspective that changing the PEG content does indeed change mRNA delivery and functional biodistribution for ZALs. And with that, I would like to acknowledge my lab. We have a pretty big lab here at UT Southwestern. And also the people you see highlighted or bolded, they had a really big part with this project. And also like to thank my PI, again, Dr. Daniel Siegert. And with that, I would like to thank you all for your attention, and I'll take any questions you may have.
Thanks, Just. Another wonderful presentation. We really enjoyed it. So to start with the last part, so about your ZAL LNPs, So you mentioned in the final part that you skipped the in vitro assays and that you moved directly to the mice. And to me, that makes completely sense. But what can one learn from the in vitro experiments that you skipped? Because to me, it's all about the delivery, right? It's all about where, in which organ does it end up. So what to do with the in vitro experiments?
So in the case of had we done in vitro experiments, we may have seen actually a lower acetate may have still been lower as opposed to the parent compound. And actually, thankfully, my boss, my PI kind of told me, he's like, hey, that's not necessarily the case, go do it in mice. So if we done in vitro, we might have actually like not done in vivo based on those results that we saw, because these are kind of new derivatives. In addition with that, looking at the acetate wild may be working better, doing some more lit review studies, so we saw that acetate is actually really important for ethanol alcohol metabolism. And when it's actually decomposed, it actually goes to from ethanol into acetate first and then processed. So in that regard, in vitro wouldn't have necessarily shown us that. We wouldn't have had the biochemical process being shown. So that's kind of the two reasons why we could definitely go back and do in vitro. But at the end of the day, we want this to be in actual like real models. So if we can do it in the mice and it shows really good results, it's kind of the main goal.
Yeah, sounds logical. Another question about the targeting. So I think you looked throughout your presentation in 3 main tissues, spleen, liver, and lung, I think it was. So as in the previous presentation, for the people that are working on muscle disorders or on brain disorders. So how easy is it to modify these 3 compounds and to direct delivery into muscle, heart, whatever other tissue?
So it's definitely possible to modulate formulations for targeting. Our lab actually developed this technology called SORT, where we add a 5th component and we actually can target to the lungs and the liver and the spleen. That being said, we have not looked at any other organs. Well, we saw them on the IVIS or AMI imaging. There was not much there. However, I will say that was with more systemic delivery or injections. In theory, one may be able to do a local injection and we may see delivery to maybe muscle or maybe also maybe in the brain as well if we did that. But at the moment, we're still just really looking at systemic delivery. But it's definitely something that is probably possible with LNPs to be able to do that.
Okay, and then about the cargo. So I think you talked mostly or only about mRNA delivery.
Yes.
Is this also possible with siRNAs, uncensored oligonucleotides, so smaller compounds? And on the other side, what is the upper limit? So what is the maximum number of kilobases or whatever you can deliver?
Yes, so on the smaller side with siRNA, so actually initial formulations were made for siRNA and they were modified to do mRNA. So I've actually gone back and done some siRNA studies as far as encapsulation goes, and encapsulation is still really high across the board. So that's definitely something that's easily doable, I'll say. And on the higher end, we have actually not fully tested that to where its farther extremities is. That's something we definitely can do. Just trying to deliver really just big cargo and see what happens. But that's not something we've done at the moment. But we believe that is definitely something that the ZALs can do.
Just for me, what is a typical compound that you deliver? So what's a typical size you are delivering right now?
Typically with mRNAs, like 2,000 bases or so. We normally get them made for us. We use luciferase mRNA to actually do the studies. That gets made by a company for us. And I think they're about 2,000, or 2 kilobases, I think, about that size.
Okay, and then last question. So which of the 3 components is your favorite component? Is it the salt, the PEG, or the cholesterol?
So as far as favorite, I would say for the experimental procedures, I would just say ZALs, only because I actually synthesized those myself. I have to do all the chemistry, start from scratch, do all the derivations. So that's probably my most favorite and my most hated too, because like it's really hard to purify, I found out. And then the other 2, PEG is interesting, I would say though. Cholesterol is pretty standard, but PEG is interesting because it's such a low percentage, even at 0.25%, we still see the liver. So that's really cool. Because as we know, we see our bodies can actually make antibodies to PEG. So we can actually have decrease that peg amount and still get good activity. That's definitely something that can be translated, we hope, in making different vaccines, cancer therapies, many therapies. So yeah.
Cool. Thanks a lot. And I give the word back to